Hello, everyone, and welcome to today's webinar titled Starting at the Source, Tissue, Extracellular Vesicles, and Alzheimer's Disease. This webinar is part of the Cell Biology virtual event, and I am Susie Valdez of Labroots, and I will be your moderator for today's event. Today's educational web seminar is presented by Labroots. So let's get started. Before we begin, I want to remind everyone that this event is interactive and we encourage you to participate by submitting as many questions as you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have any trouble seeing or hearing this presentation, just click on that Ask a Question box located on the top left-hand corner of your screen. As a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits tab located on the top right-hand corner of your presentation window and follow the process to obtain those credits. I now present today's speaker, Dr. Kenneth Witwer, Associate Professor of the Molecular and Comparative Pathobiology at Johns Hopkins University School of Medicine. For a complete biography of all of our speakers, please visit the Biography tab at the top of the screen. Dr. Witwer, welcome. You may now begin your presentation, sir.
Well, thank you very much, Susie, for that introduction. It's a pleasure to be here today. And to address all of you. I understand that there have been some talks in the track on extracellular vesicles today, and I will be talking with you about extracellular vesicles that come from tissue. And specifically, I'll be speaking about Alzheimer's disease studies that my group has done recently. So as Susie mentioned, I'm at the Johns Hopkins University School of Medicine, and I'm in the Department of Molecular and Comparative Pathobiology. I also have an appointment in neurology and also in our newly founded Richmond Family Center for Precision Medicine in Alzheimer's disease. I'd like to just start by acknowledging my funding and making my disclosures. So a few pictures of my lab here, and then the funding that we have from the NIH and from other sources. And in red, I'm designating those sources that have to do something with Alzheimer's disease. I'd also like to thank my lab members and also some of our contributors. So much of the work that I'll be showing you today comes from postdocs in my lab, Yao Huang and Tom Dredonks. We also have some excellent collaborators at Johns Hopkins, as well as collaborators from around the world. And so some of the work that we are presenting today has also come from Australia. So from the labs of Andrew Hill, Leslie Chang, and Laura Vella. And so I want to today talk about Alzheimer's disease and EVs, but I've also been asked to talk a little bit about efforts that have been made in the field to ensure that we're pursuing our studies with rigor and reproducibility. A little bit of background on myself and the group that I come from. I'm in a group of about 9 PIs that we call the Retrovirus Laboratory. And we study enveloped viruses in animal models of HIV disease, but recently, of course, we've been getting into some other enveloped viruses, including SARS-CoV-2. So we call ourselves the Retrovirus Laboratory, and that's not because nobody else at Hopkins studies retroviruses, but rather because our group was the first to study them at Johns Hopkins. And specifically back in the 1970s, some of my colleagues were studying animal models. They were studying lentiviruses that infect sheep and goats. And when HIV came on the scene in the early 1980s, it became clear that HIV was related to some of these lentiviruses. So I show you here an illustration from a science paper that my former advisor published with Bob Gallo and some others back in 1985. These are electron micrographs, and they show you very clearly that HIV and visnovirus, which is this virus that affects sheep, have similar budding processes, so their biogenesis is similar, and then their morphology is also quite similar. So you can see that electron-dense shell. You can see that there's an electron-dense core that forms after release from the cell. And I'm going to be telling you a little bit more about EVs today too, extracellular vesicles. And in fact, an enveloped virus is really just a host EV that has been hijacked by a foreign genomic program. Well, one of the early manifestations of HIV disease, AIDS, was a frank dementia. This is a paper that also came out in the mid-1980s showing that that patients of HIV, those who were infected with HIV, very rapidly had HIV proteins in brain. And in fact, AIDS encephalopathy or HIV dementia was a hallmark of AIDS, especially in the early days of the epidemic. So the group that I'm with, they developed an animal model of this HIV-associated neurocognitive disease. And this animal model is the pig-tailed macaque of Southeast Asia. So just like a human being being exposed to HIV, where we don't have an evolutionary relationship with this virus, it's only been in the population for decades or maybe a little over 100 years. These pig-tailed macaques and other macaques of Southeast Asia do not have their own lentivirus, their own simian immunodeficiency virus. This means that when we infect them with two, well, we actually use two viruses. So we use a swarm that immunosuppresses, and we use a cloned virus that is neurotropic. And this leads to development of that neurologic disease. So when we infect those animals, they respond much as a human responds to untreated HIV infection. And within 3 months, we have AIDS, we have decline of T cell counts, and we also have encephalitis in most of these subjects. The encephalitis is then scored by two pathologists, and we try to understand what is causing this pathology in the brain. Now, of course, since HIV was discovered, it only took a few years to develop the first treatments. And by the late 1990s, there were combination treatments that were able to suppress the replication of the virus while also not allowing the virus to mutate around those treatments. And so that very severe dementia, the encephalitis that was seen early, soon after HIV emerged, it has largely gone away. Now, there are still cases, of course, especially in untreated individuals. But now there is a spectrum of disease or different types of neurologic disease that afflict the HIV-positive individuals. And we could see this HIV-associated neurocognitive disease as a kind of neurodegenerative disease. And this has led us then also into studies of other neurodegenerative diseases, including the one that I'll be talking about today, Alzheimer's disease. We probably all know somebody who has had Alzheimer's disease and passed away with it, or somebody who has it today. And unfortunately, this is a disease that has a massive burden on society, on the caretakers, on the individuals who are afflicted with it, and on everybody. So we could roughly divide Alzheimer's disease into two categories. The one is familial, and this is an early onset disease, and the other we call sporadic, which comes with age. And there are factors that contribute to familial disease and to sporadic disease. Alzheimer's disease is also often considered a proteinopathy, that is to say, a disease in which proteins are misfolded, proteins are abnormally distributed, proteins form into bodies or tangles that interfere with the normal functioning of the brain. And we can classify the disease into stages under a few different regimens. And one of these classifies individuals from a score of 0, which denotes normal cognition, all the way up to 6, where we've had spreading pathology throughout the neocortex. And so we find that we really need to have biomarkers of disease. In most cases, there's no indication to go in and take a biopsy of the brain, so we don't really know exactly what's happening there. Of course, imaging efforts are underway, but it would be quite convenient if we could take a so-called liquid biopsy, maybe a sample of saliva, perhaps a blood plasma sample, and understand what is happening in a particular tissue like the brain. So I'd just like to make a reference here to Dimitrios Kapogiannis, who is my colleague, who is at the National Institute on Aging in Baltimore, and is also cross-appointed with Johns Hopkins, and then one of his collaborators, Ed Goetzel. And they have a whole raft of papers on using L1-CAM, which is a receptor that can be found on neurons and other cells as well, to obtain extracellular vesicles in the periphery that have putatively come from the brain, or at least from neurons. And they've found many markers, protein modifications that are associated with Alzheimer's disease and with other diseases. So I'm going to be talking more later about what we're doing to try to extend this work, and much of this has also been done in collaboration with Dimitrios. So the extracellular vesicle, you may have heard a little bit about extracellular vesicles today, the EVs. We define them as particles that are naturally released from the cell. They are delimited by this lipid bilayer, so a piece of the cell membrane, a cell membrane that they brought with them. They cannot replicate on their own because they have no functional nucleus. And they contain a lot of different molecular cargoes. The average EV might be around a millionth or several millionths of the size of the cell that it came from. So this is rather a small particle. We usually think of them as being in the range of about 30 nanometers in diameter, all the way up to several hundred nanometers in diameter. In fact, they can even be larger than that. They can be up to tens of microns in the case of some large oncosomes that are released by cancer cells. And in fact, there's even a debate about whether a platelet is actually an extracellular vesicle. I would say it is. I think a lot of platelet biologists would disagree. They would say that it's a cell. But it does fit this definition of not being able to replicate on its own and of having come from a parent cell. And so in a sense, the platelet and perhaps even the red blood cell, which has lost its nucleus, is even an EV. So the composition, of course, is this phospholipid bilayer, but then also proteins and nucleic acids and metabolites that are brought along from the parent cell. I'm not going to dwell a lot on the different types of extracellular vesicles, but there are many. The two that you probably hear about the most are the exosome, coming from the internal compartments of the cell, specifically from the endosome, the multivesicular body, and the ectosome, or the microvesicle, that you can find budding from the surface of the cell. But there are other ways that extracellular vesicles can be formed. Why do we study them? Why do we study them in neurodegenerative diseases? Well, there are multiple reasons. They are a source of biomarkers. So they protect the cargo, they protect their internal cargo with this phospholipid bilayer. And importantly, they can be traced back to the cell of origin. So I mentioned L1-CAM, or CD171, from the work of Dimitrios Kapogiannis. And this makes them particularly useful in the liquid biopsy, because if we have a marker of a cell of origin, no matter what that cell is, a hepatocyte, a cell in the heart, a neuron, an astrocyte, any cell that has its own characteristic surface signature, that surface signature can be released on the EVs. And so we at least have a chance to say this vesicle came from this source and that one came from that source. And that's why we like to use them, or we like to develop them for the liquid biopsy. Now, of course, EVs also have a biological function, so they can signal. They can signal in several ways. One of them would be by actually transferring functional molecules, such as RNA, which has been very widely studied, and I'm going to talk a little bit about RNA today. But they don't necessarily have to transfer that cargo. There doesn't necessarily have to be a fusion event with a target cell to release the cargo. In fact, I think that a lot of extracellular vesicle mediated interactions and biological processes come when that vesicle interacts with the surface of a recipient or a target cell. It's interesting to think of the EV as a kind of signaling platform. So it's not just one molecule like a cytokine that's binding to a receptor. Rather, you have an array of molecules on the surface of the EV that can each interact with components of the target cell and achieve a kind of signaling that would not be possible with just one molecule alone. Now, naturally, all these biological functions lead us to hope that we can develop extracellular vesicles as therapeutics. But in the midst of the excitement about therapeutics and biomarkers, we also don't want to forget some of the other functions of EVs that may not be considered so exciting, but are also extremely biologically important. One of those is taking out the trash. So EVs are carriers of toxic entities from the cell of origin. So how better to release something from the cell, something that's large, than to put it into a vesicle and get rid of it. And so the trash function, I think, is very important in preventing, but then also in the progression of neurocognitive, neurodegenerative disorders. And then finally, I'll just say that EVs can also be a form of nutritive support, so trophic support. So one cell can help another cell by transferring some molecules that can be used as nutrition. Some molecular recycling that occurs. In the central nervous system, there have been a lot of studies of EVs and how they participate in crosstalk between the different components of the central nervous system. So astrocytes, which are the most abundant cell in the brain, communicate with neurons and vice versa. The oligodendrocytes are also involved in this EV communication. And then the macrophages and microglia, the microglia being the so-called resident macrophages of the brain, they can be involved in inflammatory processes and anti-inflammatory processes through the release and the uptake of extracellular vesicles. And so the proteins that are involved in some of the neurodegenerative diseases that we know about are often associated with extracellular vesicles, whether they're inside the EVs or on the surface of the EVs. I run a weekly journal club on EVs that has gone worldwide during the COVID pandemic. And today we heard from Ed Campbell from Loyola University in Chicago, and he was telling us about alpha-synuclein, a protein that's implicated in Parkinson's disease, and how extracellular vesicles can carry this protein and shuttle it. So I want to note, though, that most of what we know about extracellular vesicles in the central nervous system comes from studies of cell culture. So perhaps we're co-culturing different cells from the central nervous system. Or it comes from samples that we've obtained from patients, such as blood plasma. But, you know, if we think about the biogenesis of EVs, where any given cell is releasing these particles at all times, and also taking up particles at all times, and even displaying these particles on its surface, we realize that the major role of extracellular vesicles is probably local, so probably on the cell that releases them or neighboring cells, and not necessarily on another organ system somewhere else in the body. So we could say that extracellular vesicles are most likely to act in tissue. But how do we study tissue? So there are multiple barriers to a good study of extracellular vesicles in tissue. So where are the EVs in tissue? They would be found in the so-called interstitial space. So the interstitial space is composed of the extracellular matrix that's released by cells, and then the interstitial fluids. Are we able to obtain EVs from this interstitial space? Yes, we are. But what are we going to see? We will see EVs that come from many different cells and many different cell types. Our challenge in studying EVs in tissue, and especially if we want to take the EVs out of the tissue, is in liberating those EVs while maintaining the cell integrity. So we do not want to disrupt the cells and create artificial particles that are going to look like EVs, but were actually produced during our processing of the tissue. So this shows us that there's an importance of just dissociating the tissue very gently, and then also characterizing the EVs in a way that lets us determine whether we're studying artifacts or actual EVs that were released in the cells. And so when my lab started to work on tissue EVs, and we looked at some of the literature that had been published, we were surprised to see that many of the studies that had been published previously were taking tissue and grinding it up or mincing it very finely. So this would be a mechanical process, a physical mechanical process that is actually disrupting the cells. And it doesn't allow us to necessarily draw conclusions about the extracellular vesicles. The other thing that we noticed was that although most of the studies would look for markers of extracellular vesicles, They would not look for cellular markers. So what would not be incorporated into an extracellular vesicle? Well, one thing might be a nuclear component, right? Because the EV is coming from the surface of the cell or from an internal compartment, but probably not from the nucleus, at least not in most cases. So-called negative markers of extracellular vesicles include things like Golgi, ER, and nuclear markers. And we found that for most studies, none of these negative markers were even examined. So it was impossible to say whether the extracellular vesicles were released in a native way, so to speak, or had been produced during the processing. This is important, and I think the international community came together around 2011 and 2012 to form the International Society for Extracellular Vesicles. And I would recommend that anybody who's really interested in studying EVs take a look at ICEV, the International Society, and the efforts and opportunities that it provides. So in 2014, there was a first initiative at kind of standardizing the field, at standardizing at least the reporting that should be done when one is reporting a study of EVs. And so this early document was called the Minimal Information for Studies of EVs. And in 2018, ICEV and its community released an update to this to these standardization initiatives. And it was called MICEV 2018 because of the year of publication. And this was an effort that included more than 400 scientists. 382 of them became co-authors of the final product. And so I had the honor and also a very large responsibility of helping to co-coordinate this with Clotilde Thierry. So this was a document that took us through really every step of an EV study, from obtaining the materials to processing them to characterizing them. And I'm only going to dwell on the characterization here because that's what's relevant to what we were trying to do with the brain EVs. And what we want to do with to show each other that we have an EV preparation is, first of all, to demonstrate the presence of EVs. And we can do this by showing transmembrane proteins or membrane-associated proteins. But we don't want just membrane fragments, right? We also want to have some of that internal cargo. And so it's also good to show a cytoplasmic protein. We don't just want to show the presence of EVs. We also want to interrogate the presence of expected contaminants. And so these could be things like plasma proteins. If we're working with blood plasma, it could be something like the Tam-Horsfall protein or uromodulin if we're working with urine. And in the case of cell culture or cell sources, like tissue, we might look at some of these deep tissue contaminants, so these nuclear markers or others that we would expect to be depleted from EVs. Now, there's no universal marker here. Each study is going to find the best markers for its own purposes. But we do give some recommendations of what some of those might be. And in addition to, showing these typical markers that are enriched in EVs or depleted from EVs, we also have recommendations on quantitation, so how to get particle counts, and also how to do single particle characterization. And I will mention two of the techniques that my lab uses to do that. All right, so getting back to the brain-derived EVs. When we became interested in doing this, there was a paper that had just come out from Andrew Hill's lab and Lara Vella's lab in Australia, and what they did was a complex but very rigorous technique to separate extracellular vesicles from brain tissue, and they were even able to do this on archived, so frozen tissues. So what they did was just to very gently slice the brain. So slice the brain sample, you have to expose some surface. Then digest it with collagenase. So this is to digest those connections between the cells, release the cells, and release the other components of the tissue. And then they did several centrifugation steps to separate debris from the extracellular vesicles. And finally, they did a density gradient. So those of you who have done density gradients, you know that that this is a very rigorous process, but it's also a very difficult process. And so not just anybody in the lab is able to do that. It requires some training and some very careful hands. So we asked the question of whether we could possibly take this protocol and simplify it a little bit and even automate it by using a size exclusion chromatography step in the place of the density gradients. And we were quite aware that we might not get quite the purity that we get with a density gradient, but we hope that we could at least eliminate some of the contaminants and have a reasonably pure population of EVs. So in collaboration with the lab that first published this, we tried a side-by-side comparison. And down here on the lower right, I just want to draw your attention to this Western blot. What I'm showing here are 3 markers. So calnexin, which is one of those deep cellular markers that we do not expect to see in EVs, or at least to be depleted in EVs. And then CD63 and CD81 are 2 tetraspanins. These are proteins that are associated with or embedded in the lipid bilayer of the EV. So you can see that those 2 EV markers on the bottom there, so on the left is the brain homogenate and then also a collagenase treated brain. But on the right, you see the comparison of the density gradient containing protocol with the size exclusion protocol. And so in both cases, we have the enrichment of the lipid bilayer markers, but we also have the depletion of the calnexin markers. And so this method, then, we published this modification of the method in the Journal of Extracellular Vesicles. And the kind of icing on the cake was that this was the first paper that was published in JEV right after the 2019 impact factor was announced. So it's I think that impact factor, I'm not a big fan of impact factors, but just getting an impact factor for a young journal is a big step, and I think is a testament to the hard work that a lot of members of the community put into building this journal. And it has published, indeed, many different methods papers and also position papers that seek to encourage the best science possible in the field. So just to summarize here, what we're doing is a minimal physical disruption, a gentle enzymatic digestion, and then we do careful characterization with those depleted and enriched markers. So now we're going to go into human samples, but we might also be interested in animal models. And so we have a few other questions that we wanted to address. One of them being, what if you can do perfusion, right? So during a necropsy of an animal, could you do perfusion to limit tissue damage and perhaps get a better yield or purity of EVs? And the other is the postmortem interval. What are we able to do with samples that have been sitting around for a while? So unfortunately, with human samples, we are rarely able to get them in a reasonable period of time. So our first conclusion was that this size exclusion chromatography method was a substitute for, or perhaps in addition to the density gradient protocol that had previously been employed. Our second conclusion was that perfusion, if you can do it, so if you have a mouse model or a non-human primate model, it's a great way to minimize digestion that might occur in that brain between the time that it's harvested and the time that it can be processed into EVs. And so I'm just showing you here 3 different variations of our method. On the top, you see the perfused samples, and there's not a lot of background debris there compared with the non-perfused samples, which are shown on the bottom. So if you can do perfusion, that's great. And it also helps out with those deep cellular markers like GM130, BIP, and calnexin. It might not eliminate them completely from the mouse brain, but it does reduce them. So we do use perfusion when we can. And then finally, what about postmortem interval? So unsurprisingly, and this was done with non-human primate brain samples, we were able to see that the debris that was visible in electron microscopy was increasing over the time of that postmortem interval, which was defined as just how long that brain was sitting at room temperature before we processed it. So the earlier you can get the brain, the better. But of course, we recognize that human brain samples are not able to be processed that quickly. So now we're applying the method. So now we're getting into the proteins and the RNAs that can be harvested from the brain. So the next few slides are going to be from human brain. And this is just normal human brain. This is not specific to any particular disease. So what we observed when we did our proteomics was that when we took different fractions of the human brain, so we took the brain homogenate, we took what we call a 10K fraction, which is large EVs that are not very well purified. This is just something that we centrifuged. And then we finally took the fraction where we had applied that size exclusion chromatography to get the most pure EVs that we could. And when we went from one compartment to the other, we were able to see that there was enrichment. And I don't know if you can see these different labels here, but in the in whole membrane and plasma membrane regions, and basically anything that's involved in the secretory process, as you would expect for exosomes. And so these were enriched in the small EVs in that very pure fraction that we were able to generate at the end of our protocol. So this was a good validation of what we were trying to do. We also saw that there was other compartment enrichments, and we were interested in particular in the lower graph here. So the CNS cell markers. So what markers of neurons, of astrocytes, of oligodendrocytes, and of microglia were we able to see in the EVs that we harvested from this tissue? This is important because what we ultimately want to do, as I mentioned toward the beginning of my talk, is to apply a liquid biopsy to obtain some of these EVs. And to, I think that there have been several studies, multiple studies that have been published on neurons. There have been a few studies that have been published on astrocytes, but there are not a lot of other well-validated markers that can be used to obtain cell-specific EVs from a peripheral sample. And so this was one of the things that we attempted to do. And so we did identify several good markers that we followed up using single particle analysis. And I'll tell you more about that towards the end of the talk. All right, so after that initial study that kind of validated our approach and showed us that we were enriching the expected components, now we went on to study Alzheimer's disease and comparing these Alzheimer's disease brains with supposedly normal controls. So our study design incorporated some non-AD samples and then 23 Alzheimer's disease samples that had also been genotyped according to their apolipoprotein E. So apolipoprotein E is one of the risk factors for sporadic Alzheimer's disease. And so there are several genotypes here. There are several alleles. So there's the number 4 allele, the number 3 allele, and the number 2 allele. So the 4 allele is the one that confers the most risk for development of sporadic Alzheimer's disease. So we wanted to understand if there was a difference in the amount or the quality of the EVs that were released from brain that had these different ApoE genotypes. The first thing to look at was counts. So how many EVs are actually harvested? Are we able to recover from a certain amount of Alzheimer's disease tissue? So how do we do this? How do we count something that is as small as an extracellular vesicle, where we see that most EVs are in that range of around 40 nanometers in diameter up to, let's say, 150? There's not like there's any kind of a cutoff. The method that we used was nanoflow cytometry. So is very much like cellular methods, hence the cyto in cytometry. You're passing your sample through a channel and hitting it with a laser. But this instrument that we used is only for small particles. So that passageway has been shrunk down to a size that is just big enough to accommodate the extracellular vesicles and not cells. The reason for that is that you want to reduce the background as much as possible so that you're able to detect very low scatter or low fluorescence events. And so the background is low. The other innovation of this instrument is that the material is passed very slowly through that passageway. So in contrast with the regular cellular cytometry, where you actually want to get as many events as possible, here you're actually trying to get that material to go slowly so that you can integrate more signal while it's in that path of the laser. So we're able to measure particles down to probably about 50 or 40 nanometers. And so I show you some things that we obtained with beads here, with some standard beads, and then EVs. So we can get sizes, and we also can get counts of our populations. And so, interestingly, we did not see any difference in the yield between the AD and the control. And I'll come back to the ApoE genotype in just a moment. We did observe, however, that it depended on how we normalize these counts. So we could normalize to the amount of tissue, or we could normalize to the number of particles. So over here on the right, you can see that I have that 10K, that relatively impure fraction, and comparing it with the EV fraction on the right. The reason for that difference in the particle yield per microgram of protein is that the EVs have a lot less contaminating protein in them than that earlier fraction. And so we have a much higher particle count to protein ratio. But interestingly, we did not see a big difference, drawing your attention to the graph on the right, in the different genotypes. And in fact, if anything, although it was non-significant, there were more EVs that were harvested from the ApoE44 genotype than the others. Although, as I say, that was not significant. Now, this is a little bit different from some of the study, one study in particular that's been published that indicates that there are fewer particles that are released. However, I want to stress that this is a study that included only a small number of samples, and we also were not using exactly the same model. I mean, we're using actually human samples here and not an animal model. So our overall proteomics results from these brain-derived EVs that we were able to harvest showed that the EV fraction, the purified EVs, were the best at separating the AD samples from the control samples overall. And what about individual proteins? Well, here we found as well that if we looked at the rather impure fraction, the early fraction, the 10K fraction, and compared it with the EV fraction, the EV fraction had many more significantly different proteins in it, and thus is the more diagnostically advantageous. What were some of these proteins? We were happy to see that some of the proteins that we found to be different between the Alzheimer's group and the control group had been reported before, including tau, including the 1433 proteins. And when we looked at the processes that were involved, we also found that the findings were consistent with our understanding of Alzheimer's disease and neurodegenerative disease in general. So some of these processes would include cellular protections against oxidative stress. Neurogenesis was one of the big ones. So, of course, the flip side to neurogenesis would be senescence, of the neurons. We saw several signaling pathways that were differentially regulated in the Alzheimer's disease brain. Also, glycometabolism, which we found quite interesting, and then even interactions between cells. So this of course is also something that has to be considered in any kind of a tissue EV study. So if there are differences in the interactions between cells, this means that cells may be more or less sticky for those particles that they release, and so the amount of EVs that we harvest from a tissue may differ depending upon the disease status, and make it a little bit more difficult to interpret results. All right, so the last part that I want to share with you here is the different RNA biotypes that we were able to observe. Now, of course, we also have individual RNA level data, but I just want to focus here on the RNA biotypes and tell you, first of all, that we didn't see any big differences in the distribution. So in other words, if we were to take a whole category of RNAs, like tRNAs, yRNAs, and I've listed them over on the right because I'm sure that the x-axis is not so easy to read. We looked at many different categories of RNAs and just at the total reads that went into each of these biotypes for the different samples. And we didn't see any significant differences between the brain homogeneous, so the total input material, and the 10K fraction, which was like this fairly impure releasate fraction. And actually, we had a similar result for the EV fraction, except for one interesting finding, and that was with the mitochondrial RNA. So there's mitochondrial RNA that was differentially apparently packaged into the EVs in the AD fraction versus the AD group versus the non-AD group, and specifically there were fewer of these. So this is a finding that we're following up on and hoping to make some interesting discoveries here about metabolism, especially. Okay, so what about looking at the biotypes and how they were incorporated into the EV fraction? So in other words, not looking necessarily at the disease, but rather at the different fractions. And here we found that not unexpectedly, nuclear RNA or the mitochondrial RNA were much less abundant in the EVs than in the parent tissue. And again, I think this speaks to the relative purity of our EV populations. But there were a couple of very surprising findings. One of them was on microRNAs. So microRNAs are probably the most studied RNA type in extracellular vesicles. And there are a couple of very good reasons for that. One is that they're important biologically. But another is that they're just they're very small, so easy to be packaged. And they're very stable. So microRNAs were among the first RNAs to be found in EVs, and they've been I'm sure studied much more than any other RNA type. But what we found is that relative to the total RNA, there was actually less microRNA in the EVs than in the parent tissue, and even then in the more impure releasate fractions. So microRNAs, I think, are extremely important. They can be excellent biomarkers. They have a function, but this finding, I think, is also consistent with other findings that we've made that indicates that microRNAs are not the only molecules we want to look at in EVs. And in fact, there might be other RNAs that are much more informative in a biomarker setting. One of those is the protein coding RNAs, so mRNAs. What we find is that there's actually more protein coding RNA per nucleotide of RNA in the EV than in the parent cell. And this was very surprising to us. But I think it's consistent with these RNAs being fragments. So what we're looking at here really is a degradome. So the RNA in the cell that has been broken down and released into EV. So this might be part of that trash disposal mechanism. Getting rid of messenger RNAs that are no longer intact. Now this might not be something that is very functional in terms of what the EV can do when it goes to a recipient cell, but it's very exciting for me as somebody who's looking for biomarkers because the diversity, the information in the protein coding fraction is much greater than what we would find in the microRNA fraction. So this is, I think, another wake-up call to me and to hopefully others in the field that we should be looking across RNA biotypes and not just at microRNAs. Now, for the microRNAs themselves, we did find microRNAs that were differentially regulated across the disease. Disease versus control, and especially some of the ones in the miR-132 family. And we could see this in the 10K fraction, but also in the EV fraction. And I'm going to leave it at that so that I can just briefly touch on one other technology that we've been using. And so I mentioned earlier that we had found some new markers in EVs in the brain that we would like to follow up in the periphery. So markers that we could use to pull out microglial or macrophage-specific EVs, markers that we could use to pull out neuronal EVs that might not have been studied before. So we're using this technology that's called single particle interferometric reflectance imaging sensing. And you can see the chip here. So this is a silicon dioxide coated chip onto which antibodies, or you could use other affinity reagents, to be printed. And so each of these little dots of antibodies or lawns of antibodies will then capture a particular particle type. So we used several capture antibodies in the chip that I'm showing you here, including several general EV markers, and then also several markers that we expect to be on neurons. After the particle is captured onto that antibody lawn, we can do an interferometric-based imaging. And this interferometric imaging will give us the diameter of the particle, and then also we can count how many particles have been captured by each of the affinity reagents. Another modality with this instrumentation is that we can then go in with up to 3 fluorescently labeled antibodies and assess the degree of signal, the positivity that might come from any of those 3 on the specifically captured particle. So this is a true single particle analysis technology that lets us say antibody A is capturing this particle, and then we also see these other markers on the surface. And I think what was very revealing to us is that we found that there was actually not a lot of L1 cam that we could detect on the surface of the brain-derived EVs. Now, that doesn't mean that L1 cam is not there; it certainly is. But there might be other neuronal markers that could be even better than L1 cam at allowing us to capture those neuron-derived EVs in the periphery. And this is also consistent with some bulk measurements that we've done using some of these same samples, using, for example, the mesoscale multiplexed protein assay. So we do have some other markers that have been found to be associated with the ApoE genotypes. I'm not going to talk about them right now, but we're pursuing what we've just found here, and I'm just going to just a few conclusions before we get to the questions. But we were able to obtain brain-derived EVs that we believe are of acceptable purity using this size exclusion chromatography method. And this is because it's partially automatable, the SEC portion of it anyway, we can process a lot more samples this way. We do want to stress that factors including postmortem interval should be taken into account and reported too in any study of any tissue-derived EVs, but especially in the brain. We were able to see that our separation protocol did yield results that were consistent with the pure EV population. And we were able to identify some markers that may be as good as or better than the field standards right now in identifying cell-specific EVs in outside of the brain. So I mean, it's interesting to look in the brain, but the brain was just the source. That's where we start. We want to understand what's there and then look for it elsewhere. So we have some ongoing collaborations to understand a little bit more about these ApoE genotypes that may only have subtle effects on EVs and their cargo, but we have some evidence that they're there. And then, of course, we also use cell culture models to try to understand the interactions between the different cells in the brain. So I will conclude my talk by just putting a little plug in for the International Society for Extracellular Vesicles. I talked about their standardization efforts. I've been serving as the chair for science and meetings for the society for several years. And if you have interest in learning more about EVs, or if you would like to host a workshop maybe on a particular EV topic, please get in contact with me. And we also have a subcommittee that's called the Rigor and Standardization Subcommittee, where we have multiple task forces that have been formed. So this is also an opportunity for members of the community to say, you know what, I don't see anything about brain-derived EVs. I'd like to start a task force on that. And there's an opportunity on the ISEV webpage to get involved in that way. So thank you again for inviting me to speak here, and I'm happy to take any questions that you might have.
And thank you, Dr. Witwer, for that informative presentation. And we will now start the Q&A portion of the webinar. If you have any questions you want to ask, please do so now. Just click on that Ask a Question box located on the far left of your screen. So let's get started. We already have some incoming questions from our audience members. Dr. Witwer, are blood-isolated bEVs emerging as a novel player in the study of Alzheimer's disease? And are bEVs considered as a blood-based biomarker of Alzheimer's?
So the answer to that is yes, absolutely. And I think there are many academic labs, but then also many companies that are working on this. So if there's something to be found, I think it will be in the next few years. If it hasn't been already, I think that, we have this question about specificity. So there are very few molecular markers that are truly specific to any one cell type. And that's a biological reality that we have to deal with.
Oops, it looks like he accidentally just stepped out of the studio, and we're going to try to get him back in here. I apologize to the audience members. And hopefully he will be right back in. Sometimes we have these audio glitches, and so we'll try to figure out what is happening. Hold, please. I do apologize. He was great. He's got this wonderful, calm voice, huh? So I'm sure he'll be right back in. Every once in a while we get bounced, so I do apologize. And I do want to remind you that if you have any questions you want to ask, this is a good time to go ahead and enter them into our Q&A box. You can find that up at the left-hand corner. Looks like already many of you have figured that out. So go ahead and add that in there. And I'll give him another minute, and hopefully he will come right back in here. And if you haven't had a chance, make sure you go to Labroots.com and check out some of the other conferences that are going to be available this month. We have so many amazing conferences that are being held here on the Labroots site. It's a great site, social media for all doctors, researchers, anyone in the medical field. So it's important for you to take advantage of that opportunity. And it looks like he is back in. We just need him to connect. Let me just connect him. You must. I'm sure he's going to get right in here. So yeah, take a good look at the different conferences that are out there. You know, Labroots does connect, you know, people in the medical field with all kinds of wonderful conferences. So you can register and, you know, join Labroots. So that way you can get updates and find out about other things that are happening. Okay. It says this is the presenter. Yes. All right, it looks like he'll be in here. He should be right back. Oh, there he is.
Oh, yes.
Go ahead and
apologize.
continue.
It seems that the connection cut out for me.
No worries. And we have such a great audience that all of them remained here. So
okay, well, thank you very much.
I don't know. All here still.
Did my previous answer come through?
I'm sorry?
The question about the novel players in Alzheimer's disease, were people able to hear that one, or should I go back to that?
Yes, we heard that.
Okay.
And then the second part of that one is it difficult to distinguish among EV types simply on the basis of protein markers or size alone? And again, before you answer that, I just want to thank our audience for being patient and staying with us. And they've already started asking additional questions, so we appreciate that. Welcome back, Dr. Witwer. Let's go for it.
All right. So size, no, I don't think that there is anything that we can do that will. Well, I shouldn't close the door completely on that. I mean, I mentioned the large oncosomes. When you see a very large EV, there's only a certain number of cell types that it could even give rise to that. But I think when we're talking about healthy cells, or reasonably healthy cells, I don't expect that we're going to be able to say where a particle came from just by its size. And that's why we need to have these molecular signatures. And ideally even more than one. And so moving towards a more single particle kind of analysis is important. So something that I often say, and I think Clotilde Terry says this too, is, we're kind of where immunology was in the 1970s with EVs, where, it was possible to look at some markers on the surface of an immune cell back then, but it was only with the development of multi-parametric flow cytometry that we could really understand the different subtypes of immune cells. And in the same way, I think that we need to be able to do that now for EVs. Of course, it's much harder because there's so much less surface area. But to the extent that we can, if we can understand phenotypes, that will allow us to understand what cell has the EV come from, and then also maybe some characteristics, something about the health of that cell.
Thank you so much. And what is the most common EV separation technique? And what are considered the most broadly applied characterization methods besides single particle tracking?
Well, that's a great question, and I can refer you to a publication that just came from ICEV, and it was published in the MDPI journal called Cells. So the first author is Royo, R-O-Y-O, and so we did a survey of the ICEV community. And what we found was that ultracentrifugation is still the most widely employed method for EV separation. Now, every EV separation technology has its advantages and disadvantages. The disadvantage of ultracentrifugation is that it doesn't result in a really highly pure product, and it can also result in damage to the extracellular vesicles. So depending on what you want to do with them, if you want to do functional studies, molecular characterization, it might not be the best method. But there have been numerous advances in the past decade. I mean, there's been a lot of changes. So what we're seeing is that there's a lot more size exclusion chromatography being applied. Of course, a very old technique, but now being applied to EVs, and other types of chromatography. So where you can just use a matrix, an affinity matrix, a sizing matrix, a combination of different matrices to achieve a very gentle separation. So I see that matrix-based bind dilute chromatography or traditional chromatography would be the kind of way that you can get your EVs in a reasonably pure fashion without harming them. And as for characterization methods, so I think that protein-based characterization is still at the top. So Western blotting or proteomics, whatever it is that you're doing to understand what proteins are there. But then you mentioned single particle tracking, yeah, so like nanoparticle tracking analysis or resistive pulse sensing to understand numbers and sizes. But we recently had a workshop on imaging of EVs, and I think seeing is believing is the old adage. And so electron microscopy is a widely used method. Cryo-electron microscopy now can really show you a lot of details about individual EVs. And then even super resolution microscopy has come into the arena fairly recently, and is really helping us to make strides in our understanding of EV biology. Oh, and I should mention
Thank you so much for your presentation today. I want to just tell you that your audience, many of them wrote in and said this was an incredible presentation. We don't have time to answer any more questions today, but I do want to thank you for your time and for your important research. And I want to thank our audience for joining us today and for their interesting questions. And any questions we didn't have time today, please know that they can be submitted all the way through the on-demand period and will be addressed by our speaker via email of the contact information provided at the time of registration. And this webcast can be viewed on demand for 6 months until March of 2021. Labroots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, stay healthy. Thank you again, Dr. Witwer.
Thank you.